Specific requirement for manufacture of active pharmaceutical ingredient Part 1. Welcome to this session on active pharmaceutical ingredient, or API, starting materials. Today, we'll break down how starting materials are defined and selected, based on revised Schedule M, paragraph 12(6), and the International Standard, ICHQ 11. Let's begin with the definition. According to paragraph 12(0.6) of the revised Schedule M, a starting material is a raw material, intermediate, or API used in the production of another API. It must be incorporated as a significant structural fragment into the final molecule. Importantly, this is the point where regulatory oversight and GMP controls begin. Selecting the right starting material is critical. As per ICHQ 11, the selection should consider the complexity of the structure, its contribution to the final API's molecular framework, the impact on impurity profiles, and the availability of control strategies, including specifications and analytical tests. The justification is the sponsor's responsibility. You must clearly explain why the selected material is appropriate, how it ensures the quality of the final API, and that impurities from earlier stages are either not relevant or are purged effectively downstream. Once defined, the starting material must be controlled. This includes specifications for identity, purity, and potency, control of the source and manufacturing process, ensuring compliance with GMP starting from this point. Globally, regulators expect full transparency. You may need to submit details of the starting material's manufacturer, synthesis steps, and quality controls. Remember, any change in the starting material may require a regulatory variation filing, based on regional guidelines. For chemically synthesized APIs, the starting material should be commercially available or produced using well-characterized synthesis steps, and positioned a few steps away from the final API, to allow sufficient opportunity for purity control and impurity removal. In summary, identifying and justifying a starting material is not just a scientific decision. It's a regulatory and quality-critical step in drug substance development. Follow the roadmap from ICHQ 11 and revised Schedule M to stay compliant and ensure patient safety. Let's now look at a couple of practical examples, one from chemical synthesis, and one involving a racemic mixture. Take the case of atorvastatin, a widely used statin drug. One of its starting materials is a lactone intermediate, which contains a key structural fragment of the final molecule. This intermediate is several steps removed from the final API and allows sufficient opportunity to control impurities and validate the process. Because it contributes significantly to the structure and is available commercially, it qualifies as a justified starting material.